thank you to everyone for joining us today. So you're probably wondering, what the heck is Health Connect South? I know what precision medicine is, but what is this other thing? So Health Connect South is a nonprofit that we started 2 years ago and started in Atlanta. And we recognized that we did a damn fine job of gathering in health industry silos in our health ecosystem. And we wanted to change that. We recognized that we had such and enjoy such a wide range and diversity and depth of health assets that if you just knew who did what and who needed what, that there was a fantastic opportunity in our health community to form these beautiful incongruencies, two things that shouldn't be together on their surface, but provided with the right platform to collaborate, can come together. And that's what we wanted to do not only in Atlanta, but throughout the Southeast. And we're very excited to hold this event here in North Carolina. And Ken, thank you very much for your help and your support in hosting us. We want to do the same in all states contiguous to Georgia. So we will be having another event in Nashville. We will be having them in South Carolina, Alabama, and Florida in the hopes to really build out this platform, to learn more about what health assets are here. We enjoy one of the richest densities of health assets in the Southeast, those states in combination, and think that there's such an incredible opportunity to do more together. And that's what we're about at Health Connect South. So I wanted to thank you all very much for being here today and taking time out of your calendars. We'd also invite you to get more involved in what we're doing. We right now tactically execute our mission in 3 different ways. 1, in-person gatherings. We have 4 of these right now a year. 2, we have a weekly radio program, which we're very excited about, that allows us to deep dive into different subjects. Precision medicine will be one coming up. Very glad to do that in person this time. And then 3, we have launched an online platform to allow for connections to be made possible online when in person is not possible. We want to build out all these platforms and want to do it with everyone's help. So please do let us know what you're working on and what you need so that we can share that with the larger health community. With that, let me introduce you to Dr. Mary Beth Thomas. She is the Vice President of the Centers for Innovation Program at the NC Biotech Center. You can read her background in the program just as well as I can. I will share with you, however, that she spent some time in the consulting world. She spent some time in the startup world, and she knows this space very well. So I think that she is extraordinarily well positioned, and North Carolina is very lucky to have her helping to garner the resources for startups, for ideas, for this space. And so with that, thank you and welcome.
Thank you, Russ and Shivani. Welcome to the North Carolina Biotech Center. We're very appreciative of Health Connect South including us in this. The precision medicine space is an area where we're becoming much more active, and we're glad that all of you could join us today. I am Mary Beth Thomas, and I work here at the center, and I am one of the main teammates here at the center that works around the precision medicine space. Much of my work and work here at the center looks at sector development, and that type of work relies very heavily on partnerships. For North Carolina Life Sciences, the Biotech Center is a convening force, but success in many of these areas relies on the engagement of many, and this is exactly what will need to happen for precision medicine to be a success. And I think that's why this type of an event held here at the Biotech Center is so suitable. I think, today's panel, or the event in general, is Precision Medicine, the Game Changer of Health. First, I really wanted to make sure that we all started with a shared description of precision medicine, and I went with the broadest level here, which I viewed as the tailoring of medical treatments based off of an individual's characteristics. And that really boils down to how I am different from each one of you at the molecular level, and how my metabolism, genetics, and lifestyle eventually make me more or less susceptible to disease. This approach to healthcare has the potential to lead to better and more effective treatments with fewer side effects, as well as providing cost savings to the overall healthcare system. The advances in our technology have allowed us to see how this is possible, but we are not quite there yet. Game changer. That is a pretty big term in my lexicon. I'm always a little leery of using that type of term. I approach it very cautiously because of how much significance it has. But it's not because it's not true, but because offering it offers lofty expectations that can lead to disappointment or frustration when it doesn't satisfy as quickly as people are looking for it to do, particularly in this case on the part of patients. But using a term like game changer is appropriate here because I think the use of precision medicine will be a paradigm shift, a shift that will move the emphasis from reactionary medicine, from reacting to a patient that is already ill, to one of prevention. Who doesn't want to be healthier in the long run? But getting to the new norm will likely be a long and somewhat arduous process given the complexity of healthcare. There are many layers that need to adapt to the shift, not the least of which are doctors, patients, and payers, meaning the insurance companies. And since technology advances are always the first wave of this shift, the first thing that will come is data. We will be awash in data, and that's where we stand today. But moving methodically through that to get to useful information that can be used to make better medical decisions will take a concerted effort on everyone's part, which is the reason that we're meeting here today. I think we will often question whether it is worth it. But even today, we can see that precision medicine is game-changing in its impact on healthcare. And now I'm going to go ahead and steal some information from the Personalized Medicine Coalition, which is a nonprofit, membership-based organization that represents innovators, scientists, patients, providers, payers. It's a national organization, and it aims to promote the understanding and adoption of personalized medicine concepts, services, and products to benefit patients and the health system. So some of their noteworthy statistics that they issued in 2014 are really amazing to me on how far precision medicine has come. Right? And as of 2014, there are 113 examples of personalized medicine drugs, treatments, or diagnostics already available on the market. From a technology standpoint, the sequencing of the genome is below $1,000 today. From the pharma industry setting, 30% of all treatments that are in late clinical development have a biomarker associated with them. 50% of treatments in early clinical development have that, and 60% of treatments in preclinical development, meaning that the industry is only using this technology more and more to better target their future drugs. From the regulatory standpoint, one of the big challenges there, but already there are 137 FDA-approved drugs that have pharmacogenomic information on their labels, and 155 pharmacogenomic biomarkers that are included on labels. From a doctor's perspective, physicians is another critical element of this ecosystem. From the electronic health records, which is the base data that all of this information will come from. In 2006, there were only 29% of doctors' offices using electronic health records. By 2013, that number was over 93%. So we're rapidly approaching that stage where our doctors' offices can pull this data in much more efficiently. And lastly, and probably the most important, is the impact on the patients and the healthcare costs. If women today receive a breast cancer genetic test of their tumor prior to treatment, there's a 34% reduction in the use of chemotherapy. If there's a genetic test that's used to properly dose a patient before they go on the blood thinner warfarin, 17,000 strokes can be prevented each year. And in just as one example of oncology, the use of it in oncology, if patients with metastatic colorectal cancer receive a genetic test for KRAS gene prior to treatment, it's over $600 million in healthcare savings. So these numbers, this is just the tip of the iceberg, right? That if this keeps advancing, this does create the paradigm shift that can lead to a much, much more effective health system. So today's panel, I think, begins to present some real-world examples of some of those representatives that are actively working in the precision medicine space. These are folks that are in the trenches. First, we have two industry representatives who are developing precision medicine tools that will be needed to make precision medicine a reality in healthcare. And as an interesting foil to their perspective, we also have a clinician who stands at the front lines of patient care and best understands what precision medicine will mean to her patients. So now I will ask each of my panelists to introduce yourselves, your organization, as well as offer maybe some of your challenges or needs that you bring here today. John?
Sure. Well, thanks a lot. Can you hear me? I probably don't even need that. But my name's John Ryals, and I'm the founder and CEO of Metabolon. It's about a 14-year-old company out near Research Triangle Park. We're about 150 people, and we've developed a very unique technology that is now becoming really an essential tool for precision medicine. And what our technology can do is we can take a blood sample from a person, and it's just a small blood draw, and in about 100 microliters of blood plasma, we'll be able to measure about 2,000 molecules. So these are small molecules that are involved in metabolism. We can measure them, we can name them, know what they are, know how much is there, and then we can fit that into maps of biochemistry, maps of metabolism. And so each human that's walking in this room maintains about 2,000 of these molecules, what we would call homeostatically. And so they're in very normal, very narrow concentration ranges. And when people get sick, these are the molecules that change. And they're changing because they're either compensating for the sickness or they are the sickness. And these can comprise even 200, there's 200 molecules that are made by your gut microbiome that are maintained homeostatically in the blood. And so we're figuring all of this out now. We've run about 4,000 studies over the years. We've been looking now a lot at the genome, and so we do a lot of work in genomics and trying to understand the data that's coming out of genomics, and we can talk about that as we go. But that's what we do at our company.
So good afternoon. I'm Al Lauritano. I'm with Becton Dickinson, and I work at our corporate R&D and innovation center here in RTP. Otherwise, I can be found usually on a United flight going somewhere. So BD Technologies, as I mentioned, is Corporate Innovation Center for BD. We have a number of early stage technology platforms that we work at, and the genomics area has been one we have been working with now for the last 3 years. If some of you have been following BD, we made an acquisition early this year or late last year called GenCell, an Irish company that is involved in not so much sequencing. There's a number of companies out there, big companies, that are doing a pretty good job. Everybody talks about how easy it is to sequence, how inexpensive it is to sequence, but what they don't talk about is the challenges with the sample preparation and collection prior to sequencing, and then there's the other challenge on the back end. What do you do with all that data? Because it's a lot. How do you make sense of it? So we decided, based upon some work we did here in RTP, that a lot of the inherent technologies within BD were, from sample collection, sample preparation, were very well suited to work at that front end of sequencing. And right now we're working with GenCell, our acquisition, to develop a front end platform to really be able to take that bottleneck out of the front part, to speed it, make it more efficient, make it less costly and less labor and expertise intensive. So that's our first foray right now into what I would consider kind of pure personalized medicine, but we're also starting to look at the back end. We've been working with startups, a number of them. We've done some incubation programs, acceleration programs, working with new technologies, at least I should say new to BD technologies like artificial intelligence, to start to then take on the challenges of the back end and what that data actually means and how it can affect a change in healthcare delivery. So I think that's enough for now.
Good afternoon, everybody. As you can see, I'm Arti Pandya from the Department of Pediatrics at UNC. I recently joined about 10 months ago as the division chief for genetics and metabolism in the Department of Pediatrics. Prior to coming here, I was and still am a physician scientist at the Virginia Commonwealth University, so not too far from here. I think this is a very exciting time and has been an exciting time for us in clinical medicine, especially for those who feel they're at the front end of translating all the discoveries and scientific advances to improve the health of patients that we see. Although I'm a pediatrician by training and a clinical and molecular geneticist, I also see adults. So I kind of feel that we are moving from the platform of whole exome, whole genome sequencing as a means to end a diagnostic odyssey, which is what we have been doing for the past 5 years at least, either as research or as a clinical practice plan, to more of what is coming down the pike, which is precision medicine in its narrow sense, which is going to involve more adult medicine, which is going to involve more of preventive medicine, and which is also going to probably combine the impact environment and our lifestyle has on our health outcomes. I think right now we definitely try as best as we can to provide the current advances in technology in personalized medicine to the pediatric population for a diagnostic odyssey, and then most likely help the management plan. But providing that comes with a lot of challenges, and I think what I'll do is just wait for questions to kind of address those challenges that we face on a day-to-day basis in our practice.
Great.
And us adults, we were kids once, too.
I forget that.
Before we leave, Artie, you used the term diagnostic odyssey twice, and could you just, being the clinician, I think you have your own set of challenges for this. Could you just explain what you mean by that?
Sure. So in this day and age, genetics has become the platform where when a multitude of specialties are unable to figure out what is going on with a child or an adult, I end up seeing them. And that means they have been to at least 3 by default subspecialists who have tried their best and done their best to come to certain conclusions about what the diagnosis is and what the management should be. And from there, we take on the challenge of trying to see if we can provide any answers using some of the more standard genetic tests, but ultimately trying to do more advanced technologic testing on these clients. So primarily a diagnostic odyssey would be where a parent has taken their child from one doctor to the other, not because they're doctor shopping, but because they're trying to identify what is the cause for their child not being healthy or well. And I would like to say that at least in 20 to 30% of these cases, we are able to offer some answers if we are able to try and get them the testing that is appropriate. So that was the first challenge, is A, getting them to the right physician, and B, getting them the testing that is appropriate in their case, which at least gives us what the diagnosis is. Once you have the diagnosis, although it doesn't necessarily mean we'll have a treatment, but those avenues are opening up so rapidly that I believe very strongly having a diagnosis will lead to a treatment much faster, because unless you know what the cause is, there is no way we can lead them to a clinical trial or some other management option.
Thank you. I appreciate that. I think as a parent, myself, I think for each one of those is a child and a family, and that's a huge aspect that sometimes gets lost as we talk about technology and insurance and all that kind of stuff. Each one of these goes down to a person or a child, and I think keeping that in the front of our mind is very important. But I think we have some of hopefully the future solution providers for these types of challenges. So, John, I really was happy to see Metabolon on the panel today. I think anytime, as was brought up earlier, anytime precision medicine gets brought up, oftentimes people go directly to sequencing. And sequencing, because it's the technology, it's improved so quickly, its cost is dropping, we're all amazed at that. But ultimately, sequencing can have its challenges. And I was wondering if you could talk a little more about your platform and how it's different from the results that come out of just sequencing. And perhaps I know you're in some indication areas. Perhaps you could use that as an example.
Sure. Well, I mean, anybody that's been dealing with exome data or whole genome data, what you become quickly aware of is there's a lot of mutations in every person. And so in the 30 million base pairs of the exome, there's at least 10,000 mutations in every person that are what we'd call non-synonymous. And so these are changes that are going to affect proteins. If you look at the whole genome, it gets to be much bigger. It's 3 million mutations in every person. And so the question is, how are you ever going to sort all this out? Because right now, the only thing that can get reported are mutations that we've already defined. And so those would be alleles that we already have got information on. And what we've been working on, and we've run about 50 or 60,000 people now, is that when we look at the blood, we can actually tell which mutations are having an effect and which mutations aren't affected. And so if you're going to get the genome sequence, then we can come in after that, or we can actually come in before it or even without it and tell you exactly what is happening in a patient. And a lot of the time we can work out a disease. In the case of undefined disease, or we were talking earlier, chronic fatigue syndrome, which is a very poorly defined disease, we can get into an understanding metabolically of what is actually driving that, and that can lead to intervention. And so once you understand what you're dealing with, you can go with groups like ARDIs that are very expert at intervention, and they can, a lot of the time, intervene and at least control the effects of the disease that the patient will have. And so we do a lot of that. We work with groups like Human Longevity, which is Craig Venter's company. We're working with Institute of Systems Biology and Arabal, which just spun out of that institute. And we work with a bunch of groups in the large sequencing centers in Cambridge, trying to actually pin down how to understand the genome, and then getting that into patients. And so we're really excited. I mean, in the area of inborn errors, we were talking about this earlier, of children, there's a lot of things we don't know. I mean, the easy to define inborn errors we can get, but you can have a patient that has a disease like argininemia, and the arginine levels, which is what's typically looked at, are all the same, but one patient, and because they've intervened successfully, except one patient's not being controlled in 3R, but all the arginine levels look the same. But when you look at what's behind that in the pathway data, you can see that that patient, he's not being controlled because there's other molecules in that pathway that are having a negative effect. And so that's the type of thing we can see. And we're now moving, actually, we're in patients now. And so we run this technology on patients. We typically are looking at patient practices like concierge medicine, and direct pay, because you don't get reimbursed for this, and so you got to go find patients that are actually going to pay for it. But we're right now pretty excited about what we're seeing.
Timing is perfect for this because oftentimes these things, John started his company how many years ago now?
It's 14.
14 years ago, and I can tell you, I was there. I knew Metabolon when it started, and most people couldn't even pronounce Metabolon. So, so I think it's come a long way. Please, if anybody does have questions, please raise your hand and offer. Donna? Donna was actually asking about the metabolomic profiles, as well as how could it affect standard of care from a cost perspective.
Right. So a couple of responses to that. The metabolome we call it the metabolome in your blood is quite constant. And so if we measure it from any 2 or 3 days, you're going to look pretty much the same. It doesn't jump. There was this assumption amongst physicians, because this is what they were taught for many years, that these things jump all over the place. If you eat something, they change. And that's just not what we see. And so, yeah, we pick up the early signatures of disease, and we actually have a product we're developing called the General Healthcare Assessment, which we can tell, are you getting diseased in about 14 different chronic diseases, and we'll pick up the earliest phase of this. So we can pick up liver disease before you'll ever see it in ALT or AST. We pick up diabetes before you're out of glycemic control. And we're doing it by picking up other areas of metabolism that aren't being screened clinically. So, and if you act on these things, and that action can be either diet and lifestyle a lot of the time, but a lot of the time it's intervention with drugs, you'll see you go back down to normal. And so, we're now working through our problem is how do we explain it to a physician, right? And so it's not actually being able to get the data. And we know what we're talking about, but it's hard to get them to know what we're talking about. And so we work a lot. That's one of the reasons we work with concierge docs, is because they spend a lot of time prepping for a patient, because typical prep time at your general practitioner is about 20 seconds. And you can tell because they pull the chart out, and then a few seconds later they open the door, and that was the prep time. And so when you want to deliver new data back, it's got to be something that doc. So I think that's what the last groups that are going to adopt precision medicine is going to be your general practitioner. But if you go to a concierge doc, they may spend 30 minutes to an hour per patient preparing for the patient, and then they spend another hour or so in consultation with them. So those are the guys that we're looking for. And these are actually, when you look at who is doing genomic medicine, it's those guys, because they got patients that'll afford to pay for it. But yeah, I think we're getting there. And I'll just give you an example. I mean, the milestone paper, or the foundational paper right now people refer to as for genomic medicine, is Tom Caskey's paper that was in PNAS a few years ago. And he had a cohort of 80 patients. They did exome sequencing. They had a medical finding on 15% of the patients that they looked at. And that's been the hallmark study that's being cited all the time. We did that same cohort and had a 25% medical finding rate. And we actually found some of his findings were incorrect. And they weren't incorrect, but he thought they had penetrant mutations and they weren't penetrant. So the patient didn't have a disease. Or in other cases, they weren't being deemed important mutations, but they were having a penetrant effect. And with Lee Hood, I think it was 80%, because Tom's cohort was a young professional cohort. Lee Hood's cohort of about 100 was a old sick cohort, and we had a finding rate of about 80% in those patients. So,
Okay, great, thank you. And please ask any questions that you have. This is meant to be as interactive as you make it. Al, let's move to you. BD is obviously, John has shared with us that his company has grown to 150 people. BD is slightly larger than that, I believe. And it has a very diverse platform of technologies. You've talked a little bit about some of those that relate to the precision medicine space. Could you talk a little bit more about that larger industry perspective, about how you are adapting to precision medicine challenges and opportunities?
So, yeah, good question. I mean, after our merger with CareFusion earlier this year, we went from 30,000 people to 45,000 people. And right now, a lot of the energy in the company is kind of focused on rationalizing and merging those two businesses, though there was a lot of synergistic opportunities there as well. But being innovative and starting new businesses within a big company has its challenges. Typically, your business presidents are rewarded on their performance of their business, and that doesn't necessarily include what they might be preparing for 3 or 5 years from now. So when BD has typically grown most effectively when they've been able to acquire an organization around that, as opposed to taking something and then trying to build it organically. That's not an uncommon model to do. So GenCell has actually given us the springboard now to create a separate genomics business unit that's based out in California, but that's not based within any of our businesses. Reports to the head of our life sciences business segment, but it is not part of the P&Ls of any of the existing businesses within that portfolio. So they do have some latitude and freedom and flexibility to kind of develop and grow and develop their products and grow a business. So, that's certainly a challenge for a big company. The other thing, though, that BD is doing that's unlike many big companies is they're really kind of embracing some of the very early stage front-end innovation. BD is not a company that's traditionally or hasn't in many years had a venture arm. So we typically engage with early stage technologies and startups in a very different way than, say, the J&Js of the world would, or the GSKs that have large VC arms. We've decided to take a much more focused and a much more active and involved way of engaging through sponsoring accelerators around the world, working and actually mentoring startup companies that are in those accelerators, where we provide BD employees to mentor those companies, provide subject matter expertise. We don't invest, as I mentioned, but we provide grants to those companies. And we also have access to outside services that we can give them pro bono as well, because these are companies that are working in new spaces. Some of them definitely personalized medicine related, others may not be, but these are areas that we feel that are going to be key to BD's future and could disrupt healthcare delivery and healthcare as we know it now. For example, we're working with the IBM Accelerator in Israel. There's 5 companies over there that we are actively mentoring. And the interesting thing about this accelerator program, IBM, their contribution to it is every company in there gets access to the Watson platform. Okay, now if you put Watson within BD, we may not know exactly what to do with that. But, well, I think we'd have some people who would know what to do with it. But in any case, but now we've got 5 companies that actually have small businesses already. And this is a matter of how can they bring it to the next level? I mean, John was mentioning the fact, what do you do with all this data and how do you make sense of it? Well, I think artificial intelligence capabilities like Watson and such, machine learning are going to go a long way to get us there. So we're starting to learn about these new technologies, new business models, as well as the technology itself. It's not just the technology anymore. For us, it's the capability and it's the business model, it's the talent, and seeing how this is going to be accepted by the customers. So right now we have a very active program, like I said, doing these accelerator programs, mentoring companies around the world. We're only limited by the number of mentors we can get within our BD organization. And these will address some of the, say, the growth challenges that big companies have as far as, instead of doing this work inside the walls of BD, really viewing each of these mentored companies as an experiment in itself. And our latest program that's kind of a next generation of that is to use that same startup garage model of taking a couple of founders and actually building our own startups. We just opened up we've opened up B Disruptive Labs in Mountain View, California. We have an entrepreneur in residence there. We're working with the faculty and staff at Singularity University, and then we'll be bringing in teams of BD people from around the world to work on specific startup ideas. Some of these will be genomics and personalized medicine related. Others may be more related to our existing businesses, but still, yet future disruptive things. As I like to say to people, and many of you know BD, we're not out there to make a sharper needle. This is all going to be, a lot of it's going to be much more digital health, data analytics, driven type of business opportunities.
Thank you, Al. And I think, BD is an interesting company because they continue to evolve how they work in this sector. I can share from my own personal experience that BD a number of years ago used to have a physical life science wing that they would incubate startup companies. I worked for one of the startup companies that BD incubated, as did Metabolon. So I think it's at a time when startup companies need that kind of support, that was BD's answer at that point in time. BD has since grown, and they haven't been able to keep the physical incubator on their campus here in RTP, but they found other ways to engage this group and work much more globally now, it sounds like, to continue to do the same type of work. Artie, let's talk with you a little bit. I think, one of the things I'm curious about is, do you as a clinician work with, I mean, obviously you just met Al and John, but do you work with companies at all within your unit within UNC?
Before I answer that question, yes. I want to kind of pick up on where my colleagues started and put it in perspective for you folks as to how we have bottlenecks that some of these technologies may be able to expedite the process for us. So the first myth out there that Mary Beth kind of started with is that a genome costs $1,000. That's really not the case. So when I have a client, patient who comes in, I have to do a trio, meaning the child and the parents, so that I'm able to interpret more of those sequence changes. And if the company is going to charge, or if the lab is going to charge anywhere between $7,000 and $8,000, which is the norm, by the time my patient or client gets the bill, it's close to $10,000 to $12,000. So that's reality check number one. If we need this technology to serve better, the cost has to come down. The second thing is the turnaround time. The turnaround time today for a whole exome that I would order out of a lab within my institute or outside is anywhere from 6 to 10 weeks. So as some of you may already know, there are 4 centers in the country that are working on the use of whole exome sequencing in the newborn period. And one of these centers is actually trying to return these results within a 2 to 2.5 week time frame. Having that kind of artificial intelligence would have made a tremendous impact. Right now, it's human intelligence. It's warm bodies that churn it out, working 10, 15, 18 hours a day. So that's the second bottleneck. If you guys out there are trying to figure out what is it that we need that kind of turnaround time to manage the data that comes out of there with a lot of validity and accuracy. So when we say artificial intelligence, all my computing friends get all excited, but I'm still very skeptical. And that is because I am the bearer of that good or not so good news. And I need to be absolutely certain that when I deliver that news and that my patient is going to act on it, it better be accurate. So that's the second part to it. The third part is, John mentioned a lot of mutations. I try to call it sequence variants, because until we know that a particular sequence change is absolutely the cause for a particular phenotype, that there is a definite cause-effect relationship, we call it a sequence variant. And by and large, in my practice, I spend more time discussing variants of unknown significance that they have received after spending $10,000. Trust me, it's not an easy thing to do. We prepare our clients ahead of time, very much saying, yes, we are all in to get an answer, but it's quite likely you're not going to get an answer. And that variant of unknown significance is something I'm trying to tell them, come back in a year, maybe we can reinterpret it for you. So how are we going to reinterpret it? And the reinterpretation comes from evidence-based medicine. And the way I'm going to get evidence-based medicine is to have very, very strong, robust databases that have that information. As some of you may know, there are many such activities going around, some at UNC, where Jonathan Berg and his group are trying to put together this with multiple different collaborators across the nation and internationally, such that this data can be curated into kind of a value that is assigned to it in terms of the confidence with which you can look at that. But it takes, we meet every Thursday for 4 hours going over mutation after mutation after mutation, assigning it a value to say, when somebody finds this, can they look at our database and say, yes, this is something that causes a disease or not? So those kind of evidence-based databases are extremely important. And also, what John is doing is, to me, a form of functional genetics. He is assigning or giving us the ability to assign function to a lot of these variants of unknown significance. And I think what is desperately needed is technology that allows us to do functional genetics. Maybe, right now we do it in a different animal model. Metabolon gets closer to the humans because we're taking the sample from the same individual that had a variant of unknown significance and then identifying what the basis of that is. So having rapid functional assays that back up our sequencing abilities that have kind of reached a zenith would be extremely helpful to us clinicians in trying to resolve some of these unknown diagnostic things. And having that, right now we have a dichotomous system. I offer it to the ones where I know it'll be reimbursed for, and I'd have to figure out very innovative ways. Some of them are research-based for the ones where I know that it is not going to be covered by insurance, and ethically, it's a big challenge for me. I mean, I do it, I offer it, but within me, I know that it's probably not the right thing to do. So having an evidence-based practice where I can convince any insurance company, third party versus Medicaid, Medicare, that this has value added to it would be helpful, and that's only possible with those databases that I talked about. And two, having the cost being a little more reasonable. And I would like to lastly say that if anybody was wondering what we did in genetics, we are a concierge medicine service because I take an hour and more to prepare for my patient, and I spend more than an hour and a half, where 50% of my time is accounted by face-to-face counseling. So that is what we do. But I hope I kind of in a roundabout way answered.
That was perfect.
And I think they set the perfect stage for me to kind of jump in and say how I use what they do to be able to deliver to the final consumer, which is our patient population.
Excellent. Thank you so much, Artie. One of the other questions, and I'm not sure if you you have a break at the 45? Yes, okay. So one of the things I just wanted to wrap up real quickly with is the partnership aspect, really. I think you've each touched on it a little bit. And could you talk from each of your perspectives, just fairly briefly, about how you feel partnerships will be important for your space within precision medicine?
Do you mean like partnerships between universities or companies, or
whatever you think will get you to the success point that you need to?
Yeah, so we partner with everybody, and so we've got about 800 companies and universities that we work with, and it's just getting data off of all these guys and then piecing it all back together that's really allowed us to go forward. And I think, you're going to have specialty. I mean, this is all evolving. We first learned we could do individual approaches about 18 months ago, and we've been evolving this since. And working with groups like HLI, this gives us large sets of humans, and so working with a group at Sanger Center in Cambridge, where we are aspiring to do 500,000 people over the next few years, and we're up to about 50,000 now. So, and we should talk, but that will give you an answer to your VUS problem. But that's how we see it.
Yeah.
So every one of our research programs at Biotechnology has partnership aspect. We're only about 150 people over 4 major research areas, genomics being one of them. So every one of those has links to external partners, whether it be startups, large companies, academia, funding agencies, as well, NGOs, government. We're going to be getting significant number of grant dollars this coming year to support a number of our work. So everything has a strong partnership. And I would say the area that I'm looking to develop most now is actually partnership with industries that, a couple of years ago we'd be scratching our head saying, why are we talking to Microsoft other than maybe some of their software, or IBM, or the same thing? But I mean, Google as well. These are the companies that are going to be moving into big time into healthcare with totally new capabilities. These are the companies I want to be working with. These are the companies that really don't care about the way things are done in the medical space right now. They're going to make their own rules. So I want to make sure they know we're open to work with them and maybe use some of our expertise in healthcare delivery to then help them then kind of hone their solution so they're addressing your needs, as opposed to saying, hey, we can sequence. What does that mean? It means there's a lot more behind that. So right now, spending a lot of time in really looking at how do we merge information technology and medical technology.
I think it's the mantra for the day, for the decade, I would say. Academics is learning fast that you cannot be an individual PI on a grant anymore. There has to be more collaboration, so that's one degree of collaboration. With industry, of course, we partner with a lot of commercial outfits that provide the testing platform we are looking for, since they seem to develop it a lot faster than some of the academic institutes, and one cannot deny it. So we definitely partner with them. I have personally partnered with small business grants with NIH, which I think is a very powerful avenue because it has a different type of peer review, which allows you to translate something that may be really helpful to the public sector in a faster way with a lot of academic peer review simultaneously. So I view it as a win-win situation with a small business grant. And yes, partnerships with industry are important as long as we are, very clear that there is no conflict of interest.
Right.
And besides that, I personally do not see any objections to it.
Thank you so much.
I'm pleased to be here today. I'm the equivalent of a learning disabled marsupial compared to the rest of this panel and the people that were before me. I don't have a background in science. Started this company from 4 years of pharmaceutical sales. I proudly boast that I never took a single math class after I graduated high school because I probably couldn't have passed them anyway. But I'm here today to talk about what we do and really break down for you a simple business that we've tried to apply, which may have some practical value. To talk about Harmonyx Diagnostics is really kind of our second company to deal with personalized medicine in a very unique space with a very limited range of tests. But to talk about that, we're owned by, our parent company is YX Genomics, and we own 4 businesses under the umbrella of YX Genomics: Transnetics, Harmonyx, Robotics, and YX Services, a lab services corporation. We run an automated genetic testing operation, and we've done that through primarily the business Transnetics, which we started in 2000, Tim Hodge and I. It does mouse genetics. We built it to supply genetic testing for researchers who are working with transgenic mice, knockout mice, knock-in mice. A fully robotic process, which is now served through kiosks, about 120 of these around the world. We process around 225,000 biopsies a month. We have about 20,000 different genetic mutations we detect, all of them on an automated platform. Biopsy tissue flies into Memphis via FedEx. Memphis is the home of FedEx, and so it's a great logistics center for us to be able to operate there. And these centers are in research institutes, so we have one at Duke, we just installed one at UNC, and they're great partners for us in helping people not spend time doing tasks in research labs with PhDs who are genotyping mice week after week because they have to do it in order to move their litters forward. So that's our primary business. Harmonyx was founded out of that business looking for ways to take that automated platform and apply it. And we were approached by a company called Medco. They asked us to participate in a 1-year program. We believed that was moving forward, and then things didn't work out the same way for the guys at Medco when they got acquired. But we were already ready. We were CLIA certified, and we took off into the world of Plavix testing, going directly to cardiologists. Because we had this pharmacogenomics platform, we'd learned that business, we could apply it. We had a medical director, and we said, well, surely every cardiologist in America would want to stop writing a drug that doesn't work 30% of the time. And so we began going into cardiologists' offices. The test was free, fully paid for by insurance. The only thing you had to do was collect the buccal swab in the office and ship it to us, and we give you the results the next day. But we learned about Dr. Semmelweis and his experience in Hungary, or in Vienna, was very similar to what we experienced in the cardiology world. Dr. Semmelweis was a physician who ran a hospital in Vienna, and where he was the chief resident. They had 2 OB clinics. One OB clinic was for the rich folks who came in, and they had a normal infection and death rate during the birth process. The other clinic was for poor people on the other side of the building. And the indigent and poor came into this side, and those people had an extremely high death rate during birth. Dr. Semmelweis began studying it because it was very troubling, and what he found was that the teaching side, the poor side of the hospital, had cadavers. And those students were working in cadavers and then going in and delivering babies. So he surmised that perhaps infection, or something like infection, but this was before Pasteur, was causing this high death rate. People in town wouldn't go in this side of the building. Women sat outside on their knees and begged not to have to give birth on this side of that building because they all knew they were going to die. There was a high death rate. So Dr. Semmelweis put some chlorine and lime outside the door of that cadaver area and had the students wipe their hands with chlorine and lime before they went in to deliver babies, and the death rate dropped 90%. But when he published the studies, he was effectively run out of the profession. The next 30 years of his life were terrible. He ended up in an insane asylum, killed himself. Dr. Semmelweis's experience was much like ours because when we went into the cardiology offices, what we found was cardiologists did not want to do the test. Not because the test wasn't valid, but because they didn't get paid for it. There was no reimbursement. There was no opportunity for them to make money. And in an industry where, frankly, physicians are starving, they're watching their revenue drop, and it was a very uncomfortable setting. So we looked at this Semmelweis effect, which is the reflex-like tendency to reject new evidence and new knowledge. All we heard over and over was, there have to be outcome studies. We need more outcome studies. Well, there were plenty of studies that proved that the drug clopidogrel is not metabolized if you have a CYP2C19 mutation in those specific areas. Plenty of studies. You don't need an outcome study to prove that. If you don't metabolize the drug's not going to turn into a drug, it's a prodrug, and you continue to write a prescription for it, you ought not do that. So after some frustration with that and realizing that we all paid $3 billion as taxpayers to sequence the human genome, and since 2001, just under $2 trillion has been paid through the NIH for all of us to find out these wonderful things that we know now about ourselves. Each family in America has paid almost $16,000 through the NIH just in taxpayer dollars for the purpose of researching all these wonderful things. Last year, or 2013, there were 10.1 million ADHD prescriptions and only 30,000 pharmacogenomic tests run on those 10.1 million prescriptions. Plavix today has 6 million people who swallowed it this morning, haven't got a single bit of opportunity for that drug to become a drug. 6 million people swallowed a pill this morning thinking that it's preventing a stroke or a heart attack, and it's not. So you either don't write the drug or do a genetic test to find out if the drug works. You can do an antiplatelet test on the backside if you want, but this needs to stop. And so we became frustrated because we feel like the pharmaceutical companies see pharmacogenomic testing as hurting sales, insurance companies look at it as more money to pay, and physicians look at it as another thing to do that they don't get paid for. So it's a very difficult spot to be in. And then in 2014, as we were in this model and rolling it forward, Cahaba, our Medicare provider in the state of Tennessee, decided it would only pay for Plavix testing in the event you had acute coronary syndrome and were getting a stent, which was 8% of the population, while they made the genius decision of continuing to pay for the drug for the other 92% of the people. And I explained and pounded on the table at the Cahaba hearings and said, how can you pay for a drug that you admit on this side 30% of the people cannot metabolize? It makes no sense. So after a while, we just got so frustrated, we decided that the one person in this equation that actually cares about whether their Plavix works and many other drugs is the patient. And so that's how our company moved forward. We decided to help try to lead a patient-centered revolution to make pharmacogenomics available to as many people as possible for as many drugs as possible for the lowest price possible, because we run 200,000 biopsies a month and we run them for an average price of about $10. So you shouldn't be taking science and making it $3,000 for an ADHD test when it's something you can do very inexpensively. So let me take one moment and show you how this model ended up. Show you this quick video. You'll see exactly how our program moves now.
Jenna and Stacy are best friends. They do everything together. They eat together. they work together, they exercise together, they even take the same medications. But as alike as they are, Jenna's prescription isn't working, and she doesn't even know. So when they came in to pick up their medication, I told them something most people don't realize. Your genetic makeup can keep a prescription from being effective. And if your doctor hasn't seen your prescription fail before, you'd have no idea that it wouldn't work. Now there's a test that can help. It's called Harmonyx, and I can perform it right in the pharmacy. It's simple, painless, and only takes a minute. So Jenna took the test. All she had to do was swab the inside of her mouth, and I sent the sample to Harmonyx. In 1 to 3 days, I can know whether her prescription is effective, or if I need to consult her doctor to find a new course of treatment. We helped Jenna find a new prescription, and now she and Stacy have one more thing in common. They each have a prescription that is right for them. Don't assume that one pill fits all. Ask about the Harmonyx test today to see if your prescription is right for you.
So that's our business model. It functions through pharmacies because we found the pharmacy to be very receptive to trying to help the patient make sure they're on a medicine that will work for them. We just have done a deal with Rite Aid and are now in almost all Rite Aids. We're not going to be in the state of New York for a little while, but we have another about 3,500 pharmacies that are signed up in our program and are involved in pharmacogenomic testing. And I want to clearly indicate we don't do everything because we can't, but the tests that we do are ADHD, we do for $89. We do a test for antiplatelets for $59. We have a test now for pain, which covers all the pain medicines from methadone to ibuprofen, looking at the pharmacodynamic, pharmacokinetic reactions of the patient and profiling them specifically. And this is how our results look. They bend the drugs into 3 categories: green, yellow, and red. Why? Well, we began with me being a learning disabled marsupial. I'm not a scientist, and I don't give a flip about most of the things that really smart people can understand, because I can't get there. What I did care about was, if I swallow this medicine, is it going to work or not work? That's it. And if we can boil it down to that and make it application-based, that's an opportunity to actually give people a chance to use pharmacogenomics at a price they can afford and be able to employ it in their lives. We, of course, copy the physician on the results that come out of the pharmacy. The pharmacist puts the physicians, whoever prescribed the medicine, which is the reason for medical necessity, they put their NPI number in. We communicate directly with the physician what the results were. The test, it's a buccal swab. It's affordable, accessible, and actionable, because we do believe people's DNA matters to them. It may not be in the healthcare environment we want it in yet, but there's at least one way to apply this to science today. So that's our business. And I'll take any questions if you have any, or if you want to wait till the end, Shivani, let me know. Yes, ma'am.
I'm curious, so if you get the test and then it goes back to the doctor, what is the response from the doctor at that point?
Well, it varies. Some doctors call us up and say, I didn't know this was available like this. This is great. Can you send me some brochures? I'd like to have them in my office. Some doctors call us and say, how dare you do a genetic test on one of my patients that I didn't order? They usually send those to me, because I'm the angry person in the office when it comes to all that. And so I used to have to I talked with them, try to talk them down. They've turned us into medical boards. I've been turned into, probably in the southern half of the United States, we've been turned into 3 states' medical boards for testing patients without the consent of the doctor. The doctor, we have a doctor. The doctor orders the laboratory test based on medical necessity. We have a board licensed pharmacist who recommends the test to the doctor. It's just not that doctor. And they'll call him their patient, which I usually challenge and say, I'm sorry, they're a patient, they are not your patient. You cannot own them, and you cannot restrict them from going to a pharmacy and buying a product that they think helps them. Yes, ma'am.
So it's not reimbursable through insurance?
We did that, but when Cahaba and almost every MAC in America changed their policy on Plavix, I surmised, all right, because I'm really smart, that if you don't care about a $55,000 heart attack on a test that was reimbursed at $297 in those days. $297 for a Plavix test. If you're not going to reimburse $297 for a Plavix test to prevent a $55,000 heart attack or stroke, you are not likely the geniuses who are going to care whether that 10-year-old child takes Strattera, then Vyvanse, and then eventually ends up on Adderall. If you don't care about a stroke or a heart attack, you probably don't care whether or not that person's statin causes them myopathy. So I didn't see how healthcare was going to start paying for something when a board of experts sat down in 2014 and decided they weren't going to pay for heart attacks and stroke prevention. I may be wrong, and down the road it may come back, but we withdrew from the Medicare program, canceled all of our insurance contracts, and do this for cash in a pharmacy because people will buy this. People want to know if the drug they're going to swallow is going to work, but the insurance company may not want to pay for it.
So is the cash price the same to?
That's $89 for the ADHD test. It's $59. We don't bill insurance.
So you just pay the pharmacy?
They pay at the pharmacy. Okay? Great. Thank you very much.
Good afternoon, and thank you for the invitation. So, yes, MITRI, which is a family health history-based risk assessment and clinical decision support tool. I'm here on behalf, really, of representing two people from the Center for Applied Genomics and Precision Medicine. Jeff Ginsberg, who's the director of the center, and Lori Orlando, who's a clinician, and also one of the faculty members, faculty members who work on the MITRI, who leads the MITRI project. So a little bit about family health history, right? So this is kind of, it seems like a step back. You've been talking about genetics, you've been talking about metabolomics, and here I am talking about family health history. But this is a quote, if you can look at, from Francis Collins from 5 or 6 years ago. That says, "Family history is one of the most powerful ways to personalize medical care. It is the bedrock of the way we predict disease and the way we keep people well." So when you stop and think about it, your most common human diseases really roll together your genetics, your behaviors, and your environmental exposures. And those are things that you typically share with all of your family members. And knowing family health history actually is probably the least complex genetic test that you'll ever do. An example of this, right? So if you have a family member who develops coronary artery disease before the age of 50, then your Framingham risk score for that disease or that condition is double. If you have a first-degree family member with type 2 diabetes, then you have 3 times the risk of the average person in the population. So just knowing family health history actually creates a lot of value in your personal health care. The other thing about family health history is it's really, it's not scary to anybody, right? So it's not like presenting a patient with the option or perhaps the need to have a genetic test, right? It's kind of a low bar sort of introduction to family history, to genetics and precision medicine. So the whole MITRI program really was birthed out of something called the Genomedical Connection, which was a research program with federal funding that was really designed to introduce the whole concept of genomic medicine into primary care. So this is really bringing it into the forefront of healthcare, not executive health, but really into primary care practices. And the goals for the whole program, and these have become the MITRI goals because MITRI was really the tool by which this was meant to happen, was really to facilitate precision medicine in clinical practice, again in the primary care setting. It was to enable risk stratification using family health history and some clinical variables, personal history included in that. It was also to enable the uptake of evidence-based risk stratified, uptake of guidelines that were with risk stratification for each individual patient so that each person got the right screening and preventive care that they really needed. And a couple of examples of this are breast MRI. It's expensive, The average woman over the age of, I think it's 50, the guidelines are changing, will be told to have a mammogram every year. But some patients, those perhaps who have an increased incidence of the disease in their family, may be better served by a breast MRI. But they are expensive, so you want to use those judiciously, and they can have some adverse effects if downstream. So you want to make sure they're targeted to the right people. Lynch syndrome is a hereditary colon cancer syndrome, and it certainly increases the risk of colon cancer. The screening and preventive options for somebody with Lynch syndrome are far different from those of the rest of the average people in the population. So the other goal of the Genomedical Connection and now MITRI is to really encourage learning and shared decision-making between patients and providers, and to provide the education that's needed just in time. So it's not, here's a whole packet of information, learn it, and we'll assess you on it later. It's as the patient's inputting information, as providers seeing information, they're getting the education that is needed, and only that's needed. So taking a good family history is something that all providers are taught to do. Everybody sees the benefit of it. They know they should be doing it, but there are a lot of barriers to actually accomplishing it. On the patient side, it's education. How do they know what to collect? How do they know to talk to their family members about it? Do they actually recognize that it's important for their own preventive and screening care? And it's also accuracy. So you think about it, when was the last time you had to give your family history, right? You're handed a clipboard in the doctor's office and said, do you have any family history of this? And well, I'm not sure. And what does this mean? What is this term? I don't know. On the provider side, it's time. Time is probably the biggest barrier. Awareness, like their providers may not be aware that, well, they need three generations to really have a fully informative family history. Then they get the information, what do they do with it? How do they run the risk calculators and there are now a number of them. Once they get the risk assessment back, what do they do with it? What are the guidelines for stratified, for people with non-average risk? And it's complex. So some of these risk calculators and some of the guidelines are really not straightforward. On the health system side, there really aren't adequate systems right now for data collection. If you have a piece of paper with the patient's family history on it, you've got to go through this piece of paper. Where do you put the piece of paper? It's not in the EMR. And you need actionable information out of that effort that patients go into with inputting that information. There aren't any tools right now that interact well with the EMR that will collect this information and deliver a result back. And then there's an awareness, I think, of the population impact. There's a focus on population health. But in reality, if you are providing the right screening and preventive measures to everybody, then you are going to impact population health. All right, so the solution that was landed upon is MITRI, which is a patient-facing, patients entering the information, guideline-driven. There are no fancy algorithms back here. This is everything that, these are, tried and tested risk algorithms. It's according to guidelines that are well accepted from the NCCN, the USPTF. It provides clinical decision support, and it's a web service. So for the patient, again, embedded education, the way we see it being used is that the patient can go in and as soon as there's a life event, they can go back in and change their MITRI record. They provide information as they're completing the family health history. And we now have a Spanish version, which is great. For the provider, it offloads that data collection time. So in that 15-minute encounter time, they're not spending the time saying, do you have any history of breast cancer or any of that? That's all offloaded to the patient. They can spend the time going through the data with the patient, figuring out what needs to be done next, and making sure that the patient is on board with those next steps. It provides some visualization tools, which I'll show you. It has all of those risk calculators like Reynolds score, Framingham risk score, BRCA Pro. All of those things are running in the background. And it does the automated risk assessment. And it provides action-oriented decision support that's tied to education. So looking forward, we do want to introduce the genetic and genomic information here, and that will allow us to really refine the clinical decision support that both the patient and the provider receive. And an example of this is kind of cool. This is a paper, the PELB2 result. It's a paper that came out in New England Journal of Medicine earlier this year, I think, where they found that a woman who had a mutation in the PELB2 gene, her lifetime risk for developing breast cancer was 33% if she had no family history of breast cancer. However, if another person came in with the same genetic mutation, but she had 2 or more first-degree relatives with breast cancer, then her risk for developing breast cancer sometime in her life went to 58%. Is it going? There we go. So this is Mitri. This is the entry point. And I think, do I have a pointer? Maybe not. So what this does is at this entry point, it tells the patient really what is family health history, why is it important, how do you go out and collect the information, what are you going to be collecting, and how do you approach your family members about doing this. Circled there in the red circle is a click, you download a worksheet, so the patient can go away, call Aunt Maud or Uncle Bill, who knows everything about the family, and figure out what they need to know. So this is the first page that the patient sees, and they're asked to put in some demographic information and some clinical values like their blood pressure and cholesterol levels and so forth. We're now adapting Mitri so that it's an app that can talk to the EMR. So by the end of this year, at least at Duke, we'll be able to pull a lot of this demographic and clinical information right out of the EMR, so the patient won't have to do this right by themselves. They then move down this and start filling out the check boxes. Do you have this disease? And there are drop-down menus, so they don't see something unless they click on it. And it asks for the age at diagnosis. And it's not, I was 57 and a half when this happened. It's, were you between 40 and 50? Were you between 51 and 60? And there we go. With mouse overs, you get this just-in-time information, this education piece. So the tool links out to Medline Plus from the National Library of Medicine, and all of the text within Medline Plus is written towards a 5th grade reading level. So the patient can understand really what they're filling in. They then move on to really putting in the information about their family, and it's pre-populated with grandparents. Everybody has full grandparents. At the beginning, you get to, say whether you have whether you're adopted. So things change at that point, obviously. You can enter unknown for if you don't know what's happened with some of these people, but you get to go through those same disease conditions that you saw when you filled in your own profile. You then move on to being able to fill out your family tree. And here you can drag and drop. So if you have aunts and uncles and you know something about them, if you have cousins, brothers and sisters, you can pull all of this information in and build out your tree. You go to the results section and it presents a pedigree. And the colored-coded boxes at the top are populated with only those things that you've indicated are in your family tree or in your health history. So it's not populated with every disease known to medicine. It's only really what's relevant to you. You can choose to see this with more information, so you can see that all of the text popped up on the family tree. So it tells you what conditions this person had, what age they were diagnosed with that condition. Or you can view the information as a grid. And this is something the physicians like. So again, the horizontal axis is populated with those conditions that are indicated in your tree. And the physician can just look down and say, wow, 1, 2, 3 family members had cancer. 4 family members had this. And suddenly it's a much easier way to review the information. And then you get a report. And this is the risk report. This is tailored for each patient. So it's screening, referrals, medications, and lifestyle. The lifestyle information actually isn't reported to the physician. That's something that is really on the patient to own, I guess. But if you can see here, and I didn't bring my glasses, so I'm not sure I can read it for you. But it's couched in terms of talk to your doctor about, and in this case, it's somebody who may be at increased risk of breast cancer. And it says, this is what you should talk to your doctor about. And it's because you put this into the tool. You had 2 relatives with breast cancer, and at least 1 relative with breast cancer in both breasts. So they actually get to say, did I really put that in there? Yeah, that's true. And then it provides more information. So in this case, it may be around genetic counseling. And it says, you know, maybe you should talk to your physician about genetic counseling because it looks like there may be something in my family, breast cancer in my family. And it says what the genetic counselor may want to talk to you about. And then you can link out, there's a link here that will take you out to more information about genetic counseling. So this is a provider report, and it's much more action-oriented, right? So for the same patient, again, I can't read this precisely, but it says, recommend referral for genetic counseling. And it has links out to the guidelines to which this is connected. And these are the risk indicators for why we're saying you may want to do this. And the references for doing so, number needed to treat, and that kind of thing. So again, it's just-in-time information for the provider, and it's broken out into like broad categories: cancer, cardiovascular disease, liver diseases, et cetera. And we even have some pharmacogenomics information in there in terms of, you know, if the person's cholesterol levels or LDL levels are very high, it may say, consider intensifying the statin therapy. And by the way, there is a pharmacogenomic test for response to statins. So the technology, again, it's has tablet and touch screen function, so you can drag, drop, swipe to build your family tree. It follows all of the standards, the AHIC standards for our high-quality FHH, a lot of data standards, as you can see. And Mitri is now compliant with a platform called Smart on Fire, which enables all of the major EHRs have adopted this, agreed to adopt these standards. And it will allow for apps to be plug and play with EHRs. Allows for single sign-on and authentication. It follows strict data standards so that you can move information in and out of EMRs. So again, this is a little bit more about Smart on Fire, an open source standards-based platform to enable an app ecosystem amongst disparate electronic health record systems. And it's funded by Argonaut is the program, which is really a collaboration between some of the major EHR providers and some of the large medical institutions, academic medical centers. And we are in the, Mitri has been in various clinics in a research setting, and it's in 5 major health systems across the US at the moment, again, in an implementation study. But Duke is now integrating Mitri into its EHR, and we hope by the end of the year that there'll be a place where you can access it on your patient portal. There'll be a tab where you can go in and click, and you'll be taken out to Mitri, though you won't know it, because once you sign into your portal, you'll have signed into Mitri. And it will be able to pull some of that demographic and clinical data from the EHR. And then at the physician side, when they come in, they'll be able to click the appropriate spot in the patient's EMR, and the Mitri report will open. It'll look kind of like a Java applet inside the EMR, so they will never have to leave. They'll never have to calculate those risk scores themselves and figure out what to do. And I think I will finish there. Thank you. Yeah, absolutely.
So will that be, you said that will be available in the patient portal by the end of 2015?
At Duke.
At Duke. So if you're a Duke patient and you go into my chart, you'll be able to access that.
That's our goal. Yeah. Yeah. So it's so all of the major EMRs like Epic and Cerner. Cerner may already be there with being Smart on Fire compliant. Epic 2015, I believe, has adopted the Smart on Fire standard, so it can start to talk to apps, any app that adopts these standards. But the implementation of Epic that Duke is running at the moment isn't the 2015 version. And so we actually have health system support to their coding a lot of these APIs before, so that Duke can begin to receive app data. So, yeah.
So good afternoon, thanks for having me. Good news, we have reception coming up. Bad news is I'm standing between you and the reception. More good news is this isn't my first rodeo. I'm often the speaker before lunch or before cocktails. So I can get through this relatively quickly and we'll keep everybody on time and moving forward. So I'm going to talk to you a little bit today about CancerLinQ and why ASCO has taken on such an enormous challenge of aggregating electronic health record as well as other data of cancer patients. ASCO is the American Society for Clinical Oncology. It's the world's home for all of the oncologists, representing over 36,000 oncologists worldwide. It's headquartered in Alexandria, Virginia. And about 5 years ago, the board was very, very thoughtful in learning that we have not been learning from the majority of our cancer patients. Only 3% of cancer patients participate in clinical trials. The other 97%, and that's 3 to 5% to be fair, depending on which literature you look at. But the other 97% of the patients are managed every day. They are the patients that are swallowing those pills, that are getting the IVs, that all of that data is residing in individual silos in electronic health records and not being brought together. And we are losing this incredible opportunity to learn. We're also in this era of, as you've been hearing about all day today, the genomic medicine, testing for genomics and basing treatments based on what's going on genomically and genetically with the patient. We're moving further and further away from this anatomical non-patient specific care, i.e. breast cancer, and it's now cancers that are targeted with tumors, or tumors that are targeted based on their genomics. One example of this is lung cancer. So back in 1986, lung cancer was called lung cancer. It was one disease. When I was going through school, it was two diseases. It was non-small cell and small cell, right? You look at 2014, and right now we know that there are 7, actually probably a few more molecular drivers that are determining the way patients are responding to therapies and what they should be treated with. This essentially causes us some troubles with clinical trials, right? If you go back to that slide and you look at 7 different molecular drivers for lung cancer, and there's only a limited patient population of lung cancer patients, and you start getting down into the genomic markers or the markers of KRAS and EGFR, et cetera, that patient population begins to get down to 10, 15, 20 patients, right? How are you going to do a clinical trial in 20 patients? We've got to be learning from these patients right now. So again, I've touched on this already, that we're only learning from 3%. This slide essentially demonstrates what happens with drug trials. On the left-hand side is when a product is being studied to come to market. It's being followed very closely in phase 3 clinical trials, a limited number of patients, right? In cancer, we're actually starting to see drugs hit the market now after only being studied in about 200 to 300 patients, right? And then it gets released into the wild and it's utilized in a lot of different people, but we're not following those people. We're not catching those adverse events that weren't seen in the initial 200 patients. On the right-hand side, once it's released into the wild, we're not capturing that data from that big blue blob, which would actually go through the top of the slide if we didn't have those breaks there. That's the 97% of the patients that we're not learning from. And the patients that we see in clinical trials, not just in cancer, but across the board, tend to look very, very different than the patients that we treat every single day in oncology. In oncology, the average age of a diagnosis of cancer is about 67 years old. The average age of a patient that participates in an oncology clinical trial is about 52 to 53. It's a full decade and a half difference. The patients that are coming into the oncology clinics every single day have anywhere between 5 and could be up to 15 different comorbidities, which means they're on anywhere from 9 to as high as 20 different drugs. Patients on clinical trials who were getting these products to get to market were essentially healthy marathon runners that ended up with cancer. They look completely different. So there is this huge need for us to learn from the general patient population. And that's what CancerLinQ is all about. CancerLinQ is for oncologists by learning from the patients that we see every single day, aggregating their data that they put into their electronic health records and being able to rapidly learn from them. Now, we all know that electronic health records are not necessarily the best clinical tool, right? I've had many physicians tell me that because of projects like CancerLinQ, they will actually start to utilize EHRs more as clinical tools, because for now, they've been built as billing tools, right? Check your boxes here so you can write off, so you can get paid for your services. And they often leave out a lot of the rich information that they either keep in their head or they put in their notes, which is going to be a challenge, especially with a project like CancerLinQ, right? Because all of the adverse event information, the progression-free survival information, et cetera, is often captured in that free text note. So we're gonna have to get better with natural language processing. We're gonna have to get better with artificial intelligence that you've heard about already. I've already said what CancerLinQ is. It's being developed by ASCO. Here's really what our main goals are. We're starting at the basis of measuring quality and improving quality performance within the practices. So essentially being able to take what we have at ASCO have developed over the last 10 plus years is our Quality Oncology Practice Initiative, COPI, which is taking quality measures, having physicians look retrospectively, do chart reviews, and check the boxes that they are doing certain things to indicate high quality care. Well, we want to bring this to the forefront, right? You don't necessarily want the plane in midair looking back to see if they did all the right quality checks, right? You want to know beforehand. So CancerLinQ is going to take quality and put it in the front view on the dashboard to be able to show us that we are providing higher quality care, which should lead to better outcomes. We'll also get to providing clinical decision support tools and insights and exploration into the hypothesis generation of data. And I'll show you this in the demo here in just a moment. And let me get to the demo. Any questions while I'm pulling the demo up real quick? Because I'm going to be showing, I'm going against the cardinal rule of presentations and doing an actual live demo. So work with me here. So I have already logged in. This is hitting the live website. We have partnered with SAP, one of the world's renowned companies in technology development and software development, to build this platform. What I am going to demo today, as I said, is live. It's hitting the HANA platform live server. Patient data that you are seeing here right now is synthesized data. So for any clinicians in the room, if you see some things that don't make clinical sense, it's because it's fake data. However, we are in the process right now of extracting data from 5 of our 20 practices that are piloting this with us, which is representative of about 200,000 patients. So our informaticists are starting to go through that data right now and begin to make sense of it. So these functions that we built will begin to fire. So this is essentially the landing page, fully customizable to the user, not the institution, to each and every user. I'm logged in as a clinician. If I don't like this actionable patients tool here, I can move it up to my little favorite spot there. I can move it down. I can add tiles, what I want to look at. There are external resources and the ability to leave the system. So if I were exploring the million patients worth of data and I found an interesting, correlation that I wanted to see if any others have published, maybe case reports. Oops, I went to the wrong one. But if I, quickly click out, go to PubMed, do the search, and see what I can find in the more structured literature. So let me demo some of the functionality very quickly. Quality performance indicator. So I mentioned this is all about improving quality. We do bring in fully identified patient information into the system so we can provide quality reports back. For the demo, we've essentially took 25 of our 160 or so quality measures, made them machine readable, and you can see them broken down into, so my favorites, ones that I happen to want to look at all the time because I might be getting reimbursed by my payer. I can look at them by breast cancer, colorectal cancer, the core measures, et cetera. That's the left-hand side. The right-hand side is how I'm currently scoring on those measures based on my patients. So I'm going to take you through very quickly this adjuvant chemotherapy being received. It's essentially a measure that's been defined through the guidelines that ASCO has that says that a stage 3 colorectal cancer patient should receive chemotherapy within 4 months of their diagnosis to get the best outcomes. And that's based on the literature that's been published and the guideline that was created. You can see that I have 5 of my patients that meet the denominator criteria for that particular measure. So they have had and been diagnosed with stage 3 colorectal cancer over the last 365 days. You can see that my current score on this measure is 80%. So those of you who are math wizards, you can figure out I'm missing one, right? There's a patient that I have not prescribed chemotherapy to. The little white dot represents essentially the way the rest of the practices within CancerLinQ are scoring. So I'm not doing as well as my peers. So I want to figure out why, because I'm a physician and I'm driven by perfection, right? So I want to go in and see what's going on. This just reorients you. It gives you a little bit better definition of what the measure is. Shows me that I can't score any worse than I'm doing right now because I don't have any more patients. And if I do the right thing on the patient that I'm missing, I can go to 100% on this score. Now, historically, if on a quality measure, an administrator might come to you and say, hey, you're missing a patient, you haven't given him chemotherapy, go find out why or go find out who. The difficulty on the physician would be to try to go through all of their patients and think about, okay, which colorectal cancer patient didn't I give chemotherapy? This system, through the EHR data coming in, is essentially identifying that patient for me. You can see I have my concordant patients, the patients that I've done chemotherapy on appropriately, and then I have this patient in the actionable time frame that has not had the chemotherapy. So I want to see what's going on with this patient. So now this system has identified the patient for me that is lacking chemotherapy. But I want to see why. Why have I not given this patient chemotherapy? What this does is it gives me a longitudinal view of the patient's health record. Where, again, if I were to go into Epic or Variant or whatever to try to figure out all the different diagnoses and all the different dates, I'd have to click through multiple screens and scroll through. I've got a longitudinal view right now of what has been going on with this patient. I can scroll in, scroll out, and these are all clickable. So you can see that she's had 4 diagnoses since we've had her in the system, 2 pathology diagnoses, 2 procedures, no radiotherapy, and I gotta get over here, but if I can scroll down a little bit more, you'd see that she has no systemic therapy. Well, let's figure out why. I look at her diagnoses. Back in January, she was diagnosed with the chronic blood loss anemia, probably from her primary care physician, right? She was going in, she was fatigued, or maybe she noticed some blood in her stool, and there was a further workup. In March, she was diagnosed with their colon cancer. I can go to the procedures and see that she had her colonoscopy on March 7th, and then she actually had the surgery on her colon on March 16th. Go back to the diagnoses again, and you can see in April she was diagnosed with a pneumonia. Well, I'm not going to be giving chemotherapy to a patient that's had pneumonia, right? Or that's currently undergoing an active pneumonia because of the effects of the chemotherapy could make her sicker. So Essentially, I now have identified the patient that I was missing, as well as why I didn't give her the chemotherapy. So now if I have to do reporting, I can, oops, sorry about that. I can go back and I can actually put a note here, and you can tell I've done this multiple times, but I can just say, the patient was sick, no RX. And then when I have to report this to get my payments or to participate in COPE, I instantly have this and I'm able to report it. So that's sort of the quality piece of this. You also have the ability to look at your actionable patients. And again, you'll notice the speed of this. This is all in real time. I'm going through a Wi-Fi and it's hitting those patients and pulling them up and rendering these views almost instantly. So again, I can look at patients based on when I need to intervene on them. The system itself identifies who those patients are, so I can go directly to the physician and have them correct any quality improvement opportunities very quickly. I'm running out of time, so I'm going to show you the power of the system very quickly. These are my patients. These are fully identified patients. Again, fake, right, if I'm a physician. This is just going to show you the ability to get through and cut through data very, very quickly. So 1,500 patients that are mine. I'm going to walk you through a little bit of a silly use case. It's a breast cancer potential use case. So I have a friend of mine who calls me and says, hey, Rob, I'm going to be seeing a patient tomorrow who's a metastatic breast cancer patient. She's been on tamoxifen for 5+ years. She's developed an MDS, and I need to put her on lenalidomide. I'm hesitant. The reason I'm calling you is because a year ago I had a similar situation. I gave her the lenalidomide, and the patient developed a DVT. So I'm a little bit hesitant. Have you ever seen that? And I say, well, Bob, hold on just a second. Let me check on CancerLinQ. So I'm going to look at my breast cancer patients. I'm going to drill down on those. So I just went from 1,500 down to 305 breast cancer patients. I want to look to see what those breast cancer patients were treated with. This is happening in real time. I can then quickly identify my tamoxifen patients. So now I'm down to 70 tamoxifen patients. I want to see what else those patients were given. Cut through those 70 patients. You can see what they were given. I've got one patient that had received tamoxifen and lenalidomide. Now I want to see very quickly if that patient had any adverse events. And lo and behold, this patient that received tamoxifen and lenalidomide did have a DVT. So I did all of that while I was on the phone with my friend Bob. And I'd say, Bob, whoa, you saw it one time, I've seen it one time. I don't know if I'd give that to your patient tomorrow. And I then say, well, hold on. I still have the opportunity to look at the collective evaluation of the 1 million patients that I now have in this database. I can save all of those steps that I just did. I'm going to call this tamoxifen and lenalidomide. I'm going to save it. And I'm going to go out to my home. I'm going to go to the 1 million patient database. Give this just a second, because again, this is hitting 1 million patients and pulling all of this data that just rendered 1 million patients. I could go through and click all those steps that I just did, but I actually have the ability to run the analysis because it ported it over, and watch the right-hand side of the screen. This is cutting through 1 million patients, doing all of those steps that I just did, and there's my answer. So essentially, there were 85 patients that received tamoxifen and lenalidomide, and only 3 of them experienced a DVT. So now this changes my view of the world a bit, doesn't it, right? Before, he and I both thought it happens 100% of the time. Now, from all of this aggregate data, from the real-world patient, we begin to see that it's really probably only 4 or 5% of the time. So he's now likely to utilize that treatment. There's lots of other great functionality built in here. The ability to start identifying patients for clinical trials, right? Many of you in the room who are in the academic setting or in the patient care setting often get asked, how many patients do you see with XYZ disease? And it's always 5, 10, 15, 20, and you really see 2. This has the ability for me to go in and begin to identify where the patients exist. So stage 4 bladder cancers, I can begin to identify if I wanted to do a stage 4 bladder cancer trial. That in California, that's where the majority of them are. Being that I'm from Dallas, there's a bunch in Texas. I can drill down into the counties and begin to see where those patients are and maybe help guide where we should begin to open up some of those clinical trials. I'm going to stop there because I'm running out of time, but there's other functionality built in, and we're adding more and more functionality as we go on. We're actually working on our roadmap for 2016 and 2017 and the types of functionality that we will be adding. One of those really cool things that we'll be adding is the patient like mine being able to look at rare tumor types because you have such large numbers, begin to bring back dashboards on how those patients were treated, what they were treated with, and what their outcomes were to provide that sort of real-time clinical decision support to physicians. So with that, I'll stop, see if there are any questions. So the question is who's providing the information? Where is it coming from? How are we installing it? So yes, at this point in time, we are installing it in a practice-by-practice basis, and we put essentially an appliance into the electronic health record data, and then that data comes into CancerLinQ. It is tremendously inefficient and difficult, but we believe that the benefit that we will get from doing it is worth it, as well as we'll get to a point where we're doing it faster, and once we have the ability to form more partnerships with the EHR companies, it'll give us that ability to do it a little bit quicker. It's another question.
So you've probably heard of a company named Flatiron, which is being funded by Google. So
Yep.
it seems that they are addressing the same issues. So I was just curious if there's any overlap or like any.
So yes, Flatiron is out there. They started about the same time that CancerLinQ kind of went down this endeavor as well. Great organization, great company. They take a little bit of a different approach. I think they have the idea of a going public type event down the path, where I think this is for oncologists, by oncologists, to help develop and design and take care of the patients, improve the quality of care for patients. But they are out there doing similar things, absolutely. Other questions? Yes?
Have you had a persistence from, not the providers perhaps, but from institutions that are releasing their statistics, their data to be aggregated?
Yeah, so very good question. As we piloted this, we were very strategic in thinking about what practices and how we went about doing this in a small type community or pilot type test. Of the 15 practices that we've approached, 3 of them essentially are academic centers. And I'll be honest with you, getting through all the red tape was not easy by any means. It took a lot of work, a lot of effort, lots of conference calls, lots of airline trips, et cetera, et cetera. But we have them on board and data are coming in. We're also working with several large hospital systems, actually Cancer Treatment Centers of America being one of them. And then we have a handful of large privately held oncology practices. So it's a spectrum. And we took that approach on purpose because we knew it was going to be difficult on the academic center side, and we wanted to learn from it as we went forward. Other questions, comments? All right, well, thank you, and I think I got us out on time.
I just want to thank everybody for your time today, but most important work is ahead of us. So I hope that you will take an opportunity this evening to make a few new connections and see who else you can help today, and to bring the power of your own personal networks into others' lives. I was amazed in listening to these presentations, both our panelists and the individual presenters, and thank you all again. It dawned on me the way my children will receive their health care will be almost nothing like what I receive and what you receive. What each of us are doing to advance health is nothing short of remarkable. And I think the next generation of health leaders certainly have their hands full in absorbing what can be and pushing the envelopes to make it happen. So with that, thank each of you for being here today. Please take time, again, to meet someone new and to do something for them. We look forward to seeing you at future events. I would also like to thank very quickly both NC Bio and the NC Biotech Center and everyone for making today possible. Thank you for your time.